Good morning, everyone, and welcome. I'm delighted to have you here today. Today, we will be exploring the bacterial endotoxin test, a crucial aspect in ensuring product safety and quality. This presentation is led by Dr. S.K. Singh, Ph.D., and will be covering the fundamentals of the test, its importance, and how it is conducted. So, let's get started and learn more about this essential topic in microbiological testing. Let's start by talking about pyrogens. These are substances, often of biological origin, that can cause fever in living organisms. They do this by stimulating the release of inflammatory cytokines, which are signaling molecules that trigger the body's fever response. Now, a common and important type of pyrogen is bacterial endotoxin. This is found in the cell wall of gram-negative bacteria. The cell wall itself has a thin layer of peptidoglycan, which is surrounded by an outer membrane. This outer membrane contains lipopolysaccharide, or LPS, which is key to our discussion. Lipopolysaccharide, or LPS, consists of three main parts. First, there's the O antigen, which is a polysaccharide chain that extends outward and varies among bacterial strains. Second, we have the core oligosaccharide, which connects the O antigen to the third part. And finally, lipid A, which is the toxic component. When the bacterial cell lyses or breaks apart, lipid A is released, and it's this part that triggers the strong immune response associated with endotoxins. Understanding these components helps us grasp how bacterial endotoxins can provoke fever and inflammation, tying into our overall focus on how pyrogens influence the body's response to infection. Let's take a closer look at LAL, which stands for Limulus amoebocyte lysate. This is essentially an aqueous extract derived from amoebocytes of the horseshoe crab, specifically Limulus polyphemus. It's a fascinating biological reagent because it reacts specifically to endotoxins, such as lipopolysaccharides or LPS. Now, when endotoxins are present, the LAL reacts in a very distinctive way. It either forms a solid gel clot, or, if you're using the kinetic turbidimetric method, it causes the solution to become turbid. This change is actually the basis for detecting and quantifying endotoxins in various samples. Diving a bit into the mechanism, the LAL reaction is part of a cascade that involves three different types of serine protease zymogens. These are inactive enzyme precursors. The first is factor C, which is directly activated by endotoxins. Then we have factor B, which gets involved downstream, and the proclotting enzyme, which ultimately leads to clot formation. There's also a gel-forming protein called coagulagen that plays a key role in the final clotting process. So, in summary, the LAL reaction is a highly sensitive, specific response to endotoxins that involves a cascade of enzyme activations, culminating in gel formation or turbidity. This cascade mechanism is what makes the LAL test such a powerful tool for detecting bacterial contamination, linking back to our overall theme of ensuring safety and quality in biomedical applications. Let's take a closer look at the LAL reaction. This reaction is a key process in our study because it involves the formation of a new compound through a specific chemical pathway. What makes the LAL reaction particularly interesting is how it highlights the interaction between the reactants and the conditions under which the reaction occurs. It's important to understand these details because they help us control and optimize the reaction for better yields and efficiency. Throughout this presentation, we've been emphasizing the importance of reaction mechanisms, and the LAL reaction serves as a perfect example of this. By understanding the key steps and the factors that influence this reaction, we can better appreciate its role within the larger context of our research. Let's walk through how the LPS activates the clotting process. First, LPS actually activates factor C in an autocatalytic way, turning it into its active form, factor C. This is a key initial step that sets off the cascade. Next, the active C then activates factor B, converting it into active factor B. This activated factor B then goes on to convert the proclotting enzyme into its active form, the clotting enzyme. It's important to note that each of these activation steps involves limited proteolysis, meaning specific cleavage events that precisely activate each factor without breaking everything down. Finally, the clotting enzyme produced cleaves two peptide bonds in coagulagen, which is a fibrinogen-like molecule found in arthropods. This cleavage results in the formation of an insoluble coagulin gel, effectively forming the clot. Now, moving on to the reaction between LAL and 1,3-beta-D-glucan, which can sometimes lead to a false positive. Here, the key point is that the serine protease zymogen, factor G, is initially activated and then triggers the activation of the proclotting enzyme, mimicking the true activation pathway. So, in essence, this reaction can give a false alarm because the presence of 1,3-beta-D-glucan can activate the pathway through factor G, leading to clot formation even when there's no infection. This highlights the importance of understanding these mechanisms to interpret test results accurately. Let's take a closer look at some of the methods we use for testing. First up is the gel clot technique. This method is essential for evaluating clot formation and stability, which can give us valuable insights into coagulation disorders. Moving on, we have the photometric quantitative technique, which includes two main approaches. The turbidimetric technique measures the cloudiness or turbidity of a sample as a way to quantify specific substances, such as fibrinogen levels. Then there's the chromogenic technique, which relies on color changes resulting from enzymatic reactions. This method is highly specific and sensitive, making it very useful for precise measurements. Connecting these points to our overall theme, these testing methods are crucial tools in diagnosing and understanding various blood disorders, helping us to make accurate clinical decisions. The gel clot technique is a method we use to detect or even quantify endotoxins. It works by observing the clotting of a lysate reagent when endotoxins are present. Essentially, if there's enough endotoxin in the sample, it will cause the lysate to form a clot under standard testing conditions. The smallest amount of endotoxin that can cause this clotting is known as the labeled sensitivity of the reagent. To ensure our results are both accurate and reliable, it's important to perform tests that confirm this labeled sensitivity. This step helps us validate that the reagent is functioning correctly and that our detection method remains precise. In the context of our overall theme, understanding this technique is crucial because it provides a straightforward, yet sensitive way to monitor endotoxin levels, which is vital for maintaining safety and compliance in our processes. This technique is a photometric assay that measures increases in reactant turbidity. Essentially, it's used to gauge how cloudy the reaction mixture becomes as the reaction proceeds. This can be classified into two main types, an endpoint turbidimetric assay or a kinetic turbidimetric assay. The endpoint turbidimetric assay relies on a straightforward principle. There's a direct, quantitative relationship between the amount of endotoxin present and the turbidity of the mixture at the end of a specific incubation period. So, after allowing the reaction to develop, we measure how cloudy it has become, and that tells us the endotoxin level. On the other hand, the kinetic turbidimetric assay focuses on the dynamics of the reaction. It measures either the time it takes to reach a certain level of absorbance or transmission, often called the onset time, or it looks at how quickly the turbidity develops over time. This provides insight into how rapidly the reaction occurs, which can be useful for different analytical purposes. Both methods use photometry to detect changes in turbidity, making it a valuable technique for assessing endotoxins in various samples. It's a versatile approach that connects the reaction's physical changes with quantitative data, fitting neatly into our overall goal of accurate, reliable endotoxin detection. Let's walk through the process of preparing these standard solutions. First, we start with a 1000 EU per milliliter solution. By taking 0.1 milliliters of this and adding 0.9 milliliters of LRW, we get a concentration of 100 EU per milliliter. Next, we take 0.1 milliliters of the 100 EU per milliliter solution and add 0.9 milliliters of LRW to get 10 EU per milliliter. Then, repeating the process, 0.1 milliliters of the 10 EU per milliliter solution combined with 0.9 milliliters of LRW yields 1 EU per milliliter. Moving further, to prepare 0.25 EU per milliliter, we take 0.5 milliliters of the 1 EU per milliliter solution and add 1.5 milliliters of LRW, which gives us the 2 lambda concentration. For 0.125 EU per milliliter, we mix 1 milliliter of the 0.25 EU per milliliter solution with 1 milliliter of LRW. Continuing, 1 milliliter of the 0.125 EU per milliliter solution combined with 1 milliliter of LRW yields 0.06 EU per milliliter, the 0.5 lambda. And finally, 1 milliliter of the 0.0625 EU per milliliter solution with 1 milliliter of LRW results in 0.03 EU per milliliter, the 0.25 lambda. The goal here is to prepare at least 4 standard solutions that match concentrations of 2 lambda, 0.5 lambda, and 0.25 lambda by carefully diluting the USB endotoxin reference standard with water for BET. For example, if your lysate sensitivity is 0.125 EU per milliliter, you should reconstitute your CSE according to the supplier's certificate to get a 1000 EU per milliliter solution. Then, you perform a lot verification using the label claim assay to ensure your test is accurate. This systematic dilution process is essential because it allows us to generate a reliable standard curve, which is fundamental for quantifying endotoxin levels accurately in our samples. All right, let's delve into the acceptance criteria. As you can see, the acceptable range is not less than 0.5 lambda and not more than 2 lambda. This range ensures that our results are within a consistent and reliable window. Moving on, the geometric mean here is 0.177. Now, the geometric mean is a useful measure when dealing with data that spans several orders of magnitude or when data is multiplicative in nature. It's calculated as the antilog of the sum of the logs divided by the number of data points, which we also see expressed as the antilog of the sum of E over F. To get a better understanding of the log endpoint calculations, we look at the mean of the log endpoints, which is calculated as the sum of the logs divided by the number of replicates. In this case, the logs are minus 0.903, minus 0.903, minus 0.602, and minus 0.602, and when we average them, we get minus 0.752. This value helps us understand the central tendency of the data on a logarithmic scale, which is crucial for accurately interpreting the assay performance. Finally, we're testing 4 replicates from each concentration, which helps ensure our data's robustness. And specifically, this verification is for the new lysate lot under what's called the label claim assay. This process is key for confirming that the new lot meets the expected claims, maintaining quality and consistency in our product. Let's take a closer look at how we calculate the endotoxin limit. The formula is EL equals K divided by M now. K represents the threshold pyrogenic dose, which is the maximum amount of endotoxin that can safely cause a fever in humans. M, on the other hand, is the maximum recommended dose of the product per kilogram of body weight. Understanding this calculation is crucial because it helps us determine the safe endotoxin levels in our products. When we calculate the endotoxin limit, we're essentially ensuring that the amount of endotoxin introduced with the product stays below this threshold to prevent adverse reactions. This process ties directly into our overall goal of ensuring product safety and efficacy, especially in injectable and sterile products. It's important to grasp how these values interact because they guide us in setting acceptable endotoxin levels, the maximum valid dose, or MVD, being a key part of this. So, in summary, by calculating the endotoxin limit using this formula, we make sure our products remain safe for patient use while maintaining their effectiveness. Now, let's look at the first example. We're dealing with a product that is administered intravenously or parenterally with a maximum dose of 100 milligrams per person. To calculate the equivalence factor, or EL, we use the formula EL equals K divided by M. Here, K is given as 5 EU per kilogram, and M is calculated as 100 divided by 70 milligrams per kilogram. When we do the math, EL equals 5 divided by approximately 1.428, which gives us roughly 3.5 EU per milligram. Moving to the second example, we have a drug where the maximum recommended dose is 50 milligrams per square meter. Again, we use the same formula, EL equals K divided by M. In this case, K is 100 EU per square meter, and M is 50 milligrams per square meter. Dividing 100 by 50, we get an L of 2 AU per milligram. These calculations are crucial because they help us compare different drugs or formulations based on their potency and dosing parameters, ensuring safe and effective treatment planning. All right, let's look at how we calculate the MVD, or maximum valid dilution. The formula is MVD equals L times the concentration per milliliter divided by lambda, or lambda. Now, L is the endotoxin limit per milligram, and lambda is the endotoxin limit per milliliter for the product. For example, if we have a product with 100 milligrams in 5 milliliters, and its endotoxin limit is 9 EU per milligram, we can plug those numbers into the formula. First, we determine the total L, which is 20, because 100 mg divided by 5 ml gives us 20 mg/ml. Then, multiplying that by the endotoxin limit per milligram, 9 EU/mg, we get 20 times 9, which equals 180 EU/ml. Next, we divide by lambda, which is 0.03 EU/ml. So 180 divided by 0.03 gives us an MVD of 6,000. Now, an important point to remember is that if you pool samples together, say 3 articles, your MVD decreases because you're combining their endotoxin limits. Specifically, you divide the original MVD by the number of pooled articles. So, in this case, dividing 6,000 by 3 results in an MVD of 2,000. This adjustment is crucial because pooling affects the overall endotoxin risk, and it's important to account for that in your calculations. Understanding these calculations helps ensure that your product stays within safe endotoxin limits while considering how samples are handled or pooled. When we consider the validity of the test, the key point is that all replicates of solutions A and D must show no reaction. This indicates that the test conditions are properly controlled and that there's no unintended activity in those solutions. Next, the result of solution C plays a crucial role because it confirms the labeled sensitivity. Essentially, it verifies that our test is working as expected and that the sensitivity aligns with what we've labeled or expected. Finally, when we look at the sensitivity of the lysate in the presence of solution B, it should fall within a specific range, not less than 0.5 lambda and not more than 2 lambda. This range ensures that our sample solution isn't affected by interfering factors under the experimental conditions we've used. All these points tie into the broader theme of ensuring the reliability and accuracy of our testing process, especially when assessing potential interference and enhancement effects. It's about confirming that our test is not only sensitive, but also specific and free from interference. This slide doesn't contain enough information. That's why we're moving to the next slide. For further support, please contact us. We're here to help you with any questions or concerns you might have after this presentation. Don't hesitate to reach out. Your success is our priority. Thank you so much for your time and attention today. We appreciate your engagement and look forward to assisting you further.